Automation in clinical trials is the use of digital systems to handle repeatable tasks, move data through a study more reliably, and support faster decisions. It can appear in recruitment, consent, data capture, query management, safety workflows, lab data transfer, monitoring, and reporting. Its value sits in ordinary operational detail. For example, a lab file can move into the clinical database without someone reformatting it by hand. A discrepancy can be flagged as data arrives, rather than being found weeks later, or a dosing change can be routed to the right team with a timestamp and audit trail instead of being pieced together through email. Modern trials often draw data from many sources: site-entered data, central lab feeds, patient-reported outcomes, wearable data, and more may all be involved. If those systems stay disconnected, study teams are left to move, check, reconcile, and interpret information manually. Automation helps most when routine steps need to happen in a controlled way. Standard checks can run according to agreed rules, tasks can be routed to the right team, and systems can record who did what and when. This can reduce avoidable variation and give teams a clearer operational base while leaving judgment with the people responsible for review. Data cleaning is often a sensible place to start. Edit checks, reconciliation steps, and query routing can involve high volumes of repeated work. In query management, for example, a missing lab date can trigger a check, an out-of-range value can be sent for review, and a site can receive a prompt sooner. The review step still needs expertise. A raised liver enzyme may look like a simple data point on its own, but with dosing history, concomitant medication, visit timing, and other clinical information, it may need closer attention. Automation can surface the issue. Experience review determines what the issue means. Consent workflows are another useful area. eConsent is often discussed as a participant convenience, but it can also support version control, document completeness, confirmation that the correct version was used, and visibility of the consent pathway across the study. For remote or hybrid study activity, digital tracking and alerts can help teams see where attention is needed. Integration is where many projects become more difficult. If systems do not exchange data cleanly, or if the data model is inconsistent, teams may simply automate confusion. That is why process design has to sit alongside technical design. Teams need to know which system is the source of truth for each data type, how changes are propagated, what gets validated, and who reviews exceptions. AI may support patient matching, pattern detection, document handling, or earlier risk identification with the appropriate oversight. Teams need to understand how outputs are generated, reviewed, and how decisions remain explainable and traceable. In many trial settings, the aim of using automation is better use of expert time. All functions and operational leads still have central roles, but their work may shift away from repetitive administration and towards review, design, and oversight. For teams starting small, the first step is to map where time is being lost and where avoidable risk sits. Choose one or two processes that are repetitive, measurable, and painful enough to improve. Define ownership, success measures, validation needs, auditability, and user experience. Automation works best when it supports trial delivery, with governance, training, and human review treated as part of the work. To learn how Quanticate can support your next trial, email inquiries at quanticate.com or request a consultation through our website. Thanks for watching, and don't forget to subscribe for more practical insights on clinical trials and modern drug development.